The purpose of my project was to determine the effects of prednisone on planaria growth and fission. And planaria are like these little flatworms that if you cut them in half, they regenerate into 2 new worms. And my results were that the prednisone was able to speed up the regeneration process of the planaria, but in the end, they had negative health effects overall.
I tested the comparison between auditory-only information and visual information on auditory comprehension and retention, and I discovered that visual information had a significant increase in comprehension.
I study a collagen-19 point mutant that leads to schizophrenic-related behaviors due to a depletion in inhibitory synapses due to early stop codon from impaired protein structure.
So I wanted to find out the most effective way to extract caffeine from tea. And so I used the liquid-liquid extraction method with methylene chloride, and I used 3 milliliters of methylene chloride and 6 milliliters. And I found that the 6 milliliters of methylene chloride extracted the most caffeine from the tea.
So my project was to simulate the behavior, the arsenic absorbing behavior of a bacteria called Campylobacteria by constructing a sediment column with sediment and iron oxide mixed, and then testing how much arsenic of a sodium arsenite solution it would absorb. And I found that the iron oxide did have a significant effect on the arsenic concentration in the solution.
So for our project, we built a circuit to detect the ripeness of produce, and we did this by taking 4 different types of produce, and we tested the resistance of each. And we hypothesized that the red produce would have the highest amount of resistance, which proved our hypothesis correct.
I created interpolymer complex hydrogels that are chitosan based, and they were composed of sodium polyacrylate and polyacrylic acid, and I used them to purify water that's polluted with copper 2 nitrate, which is what I use for my model pollutant. And I found that polyacrylic acid hydrogels they had higher absorption capacities. However, sodium polyacrylate hydrogels were easier to apply, as I did not have to centrifuge the solutions after to remove the IPC from the solution.
Our project was using various sodium alginate beads to remove toxins from water. We tested them on dyes and heavy metal ions in water.
We also experimented on the disposal of said beads through concrete, and we found that they did not do anything with the structural stability of said concrete, and they can purify water.
I tested the effects of pine tar on the travel distance of baseballs. I put an equal amount of pine tar on an equal amount of baseballs and found out that the balls with pine tar did not go as far as the balls without.
My project was on the effect of AGX51 on the proliferation and differentiation of NHLF and IPF cells, which are healthy and disease cells. I conducted this experiment in Dr. Sasi's lab using DNase treatment, qPCR, and RT transcription to help determine the amount of differentiation occurring in healthy and disease cells, as well as the proliferation occurring in the cells. I did this by adding a 20 micromolar concentration of AGX51 to all differentiated cells and seeing if it decreased the amount of differentiation in the lung cells. I then conducted a treatment of different amounts of AGX51 at different micromolar treatments to see what would be most effective in stopping the effect of proliferation on the cells.
My project was the effect of neodymium magnets on plants, and I used to plant radishes and mung beans, and I extracted the chlorophyll, got the dry mass, and performed a seed germination experiment. And in that, I found that the results had no statistically significant difference.
So my project was a novel, non-invasive way to quantify hydration levels in a human skin model. And the objective of this project was to see whether there's a correlation between hydration levels and conductance, which there was. And this could improve healthcare system, and it could improve performances of athletes.
My project was what is the effect of different substances on apple browning. This experiment, I tested several different substances to determine which one was the best at stopping apple browning. My results showed that honey is the best or lemon juice is the best at stopping apple browning, and water is the worst.
So my project is the effects of different vehicle fluids on time of percolation through permeable pavement. And I discovered that power steering fluid had the slowest time of percolation with 36.5 minutes, and gasoline had the fastest time of percolation with 0.222 minutes.
My project was about magnets and how they produce levitation, and I discovered that the more magnets added, it makes it levitate higher, but along with that comes diminishing returns, which means that every magnet added adds less and less to the total height while still adding some extra height.
So I tested 3 different AI algorithms in a password guessing program to see which one would do the best. The difference between these different algorithms are the inventory size that they have. I tested 3 different ones, one of them with a larger inventory than the other 2, and the one with the larger inventory did the best job.